Since ancient times, every living creature on planet Earth has gone through evolutions of structural and cellular development to build immunity, in order to survive the ever-changing environment and to be able to prevent diseases caused by the pathogens. Like invertebrates, or animals that lack a vertebral column or backbone, such as insects, have outer shells that we call exoskeleton, made from chitin, which is a polysaccharide present in the exoskeletons of arthropods. As a result, the shells are strong and can act as an effective outer shield. Other than this, there are enzymes called lysosomes in the intestines that destroy food that is contaminated with bacteria or pathogens. And this species also possesses hemocyte cells that use a process called phagocytosis, an action of engulfing pathogens with its plasma membrane to destroy invaders such as Plasmodium, a protozoa that causes malaria in humans. Now that we know about the immune systems of the invertebrates, what about the vertebrates?
Vertebrates are animals that have a backbone inside their body. For example, humans. This is Sarah. Sarah is a human. In her human body, there is a defense mechanism to defend pathogens from entering the body. This mechanism is called the first line of defense, present in different body systems, such as sweat glands, oil glands, stomach lining, urinary bladder lining, and birth canal mucosa, have specific substance that can capture and inhibit the growth of pathogens. In trachea, a part of the lower respiratory tract, there are cilia and mucus to trap foreign particles like antigens. And as a result, those antigens will be swept out of airways and out of the body in the forms coughing and sneezing. However, what if the first line of defense wasn't able to defend all the antigens? Don't worry, that's why there is the second line of defense.
When a wound occurs, the body may be approached by the outside invaders. Luckily, we've got the Avengers. The second line of defense. Here comes the members. First ones, the brother phagocytes, neutrophil, and macrophage. Their job is to distinguish the invaders by phagocytosis. Second one, dendritic cell. Securing the surface tissue, it also engulfs the invaders with their plasma membrane and activates the specific defense mechanism. Following up, the foreigner's detector, eosinophil. Guarding under the epithelial cell, once it has detected the target, the parasites, it'll have a specific enzyme secreted to save the day. On to the next member. We have basophil, who secretes histamine to stimulate the process of the immune system. Finally, natural killer cell, who actually is a natural killer. It secretes chemicals which stimulate apoptosis, the process of programmed cell death, to keep our body remain unconquered.
Here are the body response when approached by outside invaders. Mast cells will signal macrophage with histamine for help. Once it arrives, no foreigner will be left alive. Also, basophil and mast cells have their histamine secrete to cause vasodilation, the widening of blood vessels, which is white blood cells and lymph way to the wound. This process makes the wound swell and body temperatures arise. Aha! White blood cells are here. Bind the uninvited guests and farewell the damaged tissue. The pathogen. Tissues and the warriors who sacrifice white blood cells will then turn into abscess and get eliminated through the wound. The mechanisms mentioned previously. Inflammation is non-specific defense mechanism. Then, how about specific defense mechanism? The invaders not only activate non-specific defensive mechanism, but also stimulated specific defense mechanism. Let's see how.
Third-line defense is different from the first and the second as it applies to adaptive immunity, aiming for specific targets. And the white blood cells that will play an important part here are lymphocytes and antigen-presenting cells. In this section, the term antigen is used for calling pathogens that trigger adaptive immunity. Something like criminals with a bounty on their heads. Macrophage cells will get rid of pathogens by phagocytosis. After digestion, fragments of the antigen are left. These fragments are brought to the protein on the cell surface of a macrophage called Major Histocompatibility Complex Class II, or MHC Class II. The main purpose of this process is to induce lymphocytes and other cells' activities. We call this macrophage antigen-presenting cell, or shortened as APC. To make it clear, lymphocytes are a group of white blood cells with a big nucleus that takes up pretty much the entire cell space. They can be categorized into two groups, T cell and B cell. The first, T cell, is a lymphocyte that grows from bone marrow, but later comes to full growth at thymus gland. The second one, B cell, grows and then matures in the bone marrow.
T cells have an antigen receptor called T cell receptor or TCR on their surface that is specific to the antigen. The TCR's limb is positioned to attach to the APC-presented antigen fragment. There are two types of T cells that are very important in this process: helper T cell and cytotoxic T cell. Helper T cells will capture antigen fragments presented by APC and produce a signaling chemical known as cytokine and cause other T cells and B cells to respond to pathogens. When stimulated by helper T cell, cytotoxic T cell will increase its number by cell division. And now, cytotoxic T cell is ready to fight. After encountering the infected cells, it will release an enzyme, perforin, to penetrate them, and also release granzyme into the perforin pore to stimulate apoptosis, causing the infected to pop. B cells have an antigen receptor called B cell receptor or BCR on their surface that is specific to the antigen. The BCR's limb has two areas that can attach to the antigen. When the antigen attaches with BCR and acquires cytokine from the helper T cell, the B cell is stimulated to proliferate. Some will grow in size and change into plasma cells, which play a role in generating antibodies. And these antibodies will collect antigens, for it would be easier to be disposed of by phagocytes. Other B cells will develop into memory cells, which are very helpful as they can detect and get rid of the same antigens if they were to invade our bodies again.
Sometimes the immune system in our bodies become weird and don't do what they are supposed to, which is to fight off threats. This can lead to a lot of complications such as AIDS, acquired immunodeficiency syndrome that is caused by HIV, human immunodeficiency virus, allergies, and autoimmune diseases. AIDS is a symptom that occurs when human immunodeficiency virus, called HIV, enters the body. HIV attacks a white blood cell called the helper T cell, which is the most vital part of the immune system, and takes control of the cell's DNA. It does this by entering inside the helper T cell and undergoing reverse transcription using RNA to transcribe HIV DNA. Then the new HIV DNA enters the nucleus and takes control of the helper T cell. Having been infected with HIV DNA, the helper T cell will produce more HIV proteins. In the end, the helper T cell will be destroyed, and then the virus will move on to other helper T cells until the number of helper T cells in the body reduces, making the body susceptible to AIDS.
Allergies are the body's responses to harmless foreign substances as part of the immune system responses called hypersensitivity reactions. The foreign substances that trigger allergies are called allergens, such as the pollen wreaking havoc right now or other things like insect bites. When an allergen enters the body, the B cells will develop into plasma cells, and the plasma cells will produce a specific antibody that targets the allergen. The antibody will attach itself to basophil white blood cells and mast cells. When the same allergen enters the body again, the basophil and mast cells will release a chemical called histamine. This will result in allergic reactions like eye and nose irritation, runny noses, and in some cases breathing difficulty, since histamine will make the smooth muscles in the airways contract.
Autoimmune disease happens when the immune system in the body cannot tell between your own cells and foreign cells. This will make some white blood cells, such as cytotoxic T cells, attack normal cells by mistake. Two main diseases are the result of this: lupus or SLE, short for systemic lupus erythematosus, and rheumatoid arthritis. Lupus is when the body produces antibodies that cause inflammation in various organ systems, such as the circulatory system or the nervous system. Lupus is more commonly found in women. Symptoms include red butterfly-shaped rashes on the face and nose, muscle aches, and joint pains. Rheumatoid arthritis is a long-term disease that is a result of antibodies attacking the joints and causing inflammation. Symptoms include pain, stiffness, swelling in more than one joint, and followed by inflammation.
Developing immunities for your body is one of the ways to maintain good health and prevent various diseases. There are two main types: passive immunization and active immunization. Passive immunization is when antibodies of a specific antigens are given to the body directly, causing the body to react to those antigens immediately. However, these antibodies are temporary and may not remain in the body for long depending on the amount of antigens received. The example for this type of immunity are when given from the mother during pregnancy, also through breastfeeding, and from serum extracted from animal blood such as horse. Active immunization is when the antibodies are produced in the body after receiving the antigens. At the same time, the body will create memory cells that are specific to the previously received antigens and store them. So next time, when the same antigens enter the body, those stored memory cells will be able to stimulate B cells to produce antibodies quickly. This is the reason the antibody from this type of immunity can stay in the body for a longer period of time. The example for this type of immunity are from getting infected directly and from vaccination.
SARS-CoV-2 is an enveloped virus covered by a layer of lipid with spikes coming out of them. These spikes attach to receptors on cells, and some of them are good immune system triggers. The virus spikes may be a substance or enzyme. To deal with the enormous wave of COVID, humans create various types of vaccines. But what are the differences between all of them? First one, mRNA vaccine. It uses messenger RNA protected lipid nanoparticles, which are like bubbles of lipids. This messenger RNA carry an encoding or a set of instructions to make the spike protein. Once these lipid nanoparticles are absorbed into a cell, the cell will display the spike protein, making the body's immune system respond. For example, Pfizer and Moderna. Second one, the viral vector vaccine. Use a double-strand DNA encoding for spike protein that isn't a harmless virus. This harmless virus will infect cells and display the spike protein, making the body's immune system respond. For example, AstraZeneca, Johnson & Johnson, and Sputnik V. Third one, the virus-like particles vaccine is made of nanoparticles. These nanoparticles are coated with synthetic spike proteins. An additional element called adjuvants is added with arrows to boost the immune reaction. For example, Nanovax. And the last one, inactivated virus vaccine, is made of chemically inactivated coronavirus, so it will not replicate, but all the proteins still remain intact. For example, Sinovac and Sinopharm.